The coagulation cascade occurs during secondary hemostasis. During this stage, several clotting factors facilitate complex reactions to create a fibrin mesh, which stabilizes the platelet plug that was created during primary hemostasis. As the clotting cascade initiates, clot dissolution is simultaneously triggered, but that process will be covered in a later video. The coagulation cascade can start from two different pathways, the extrinsic and intrinsic pathways. The extrinsic pathway is activated with external tissue injury to the blood vessel, while the intrinsic pathway is activated by internal vascular injury and endothelial damage. Both pathways begin independently from each other, but converge at the common pathway where factor 10 is activated to 10A and the cascade continues. The little a after its name indicates that the factor is now activated and ready to do its job. The extrinsic pathway begins with external trauma damaging the vessel walls. This exposes the smooth muscle layer of the blood vessel where tissue factor or factor 3 is found. Free-floating factor 7 in the blood comes into contact with the exposed tissue factor and becomes activated. Calcium ions, which are also floating around in the blood, bind to activated factor 7, creating a tissue factor 7A complex that is now able to cleave factor 10 and activate it into factor 10A. Interestingly, calcium ions used to be called factor 4 as a part of the cascade, but it was later on discovered that the factor 4 was just calcium ions. Factor 10A is now ready to continue on to the common pathway. But before moving on to the common pathway, let's talk about the intrinsic pathway. After internal vascular injury to the endothelial layer, collagen fibers are exposed. The intrinsic pathway is activated when a free-floating factor 12 in the blood comes into contact with the collagen fibers. Upon contact, factor 12 is activated to 12A. Factor 12A then cleaves and activates factor 11 to 11A, and while activated, factor 11A binds to calcium ions to cleave and activate factor 9 to 9A. Factor 9A then binds to more calcium ions, as well as an already activated factor 8A, to create a complex that is able to cleave factor 10 into the active form 10A. With the two pathways now connected at factor 10A, it plays a critical role for the rest of the cascade. Factor 10A cleaves factor 5 to 5A, and together with another calcium ion, factors 10A and 5A bind together to form a prothrombinase complex. This complex in turn activates prothrombin, or factor 2, into thrombin, or factor 2A. Each single complex can catalyze and activate thousands of thrombin, which is pivotal to the amplification process of the clotting cascade. Thrombin has multiple roles in the procoagulation process. After binding to a calcium ion, its first role is to facilitate the activation of fibrinogen, or factor 1, into fibrin, or factor 1A. The fibrin aggregates together to form a weak mesh over the platelet plug that was previously created during primary hemostasis. Thrombin's second function is to cleave factor 13 to 13A. Factor 13A combines with a calcium ion to form crosslinks between the fibrin strands, which strengthens the mesh to form a clot. Thrombin's third role is to activate platelets and conforming them into a shape that easily allows them to stick to other platelets over the wound, further adding to the platelet plug. The fourth role is to activate other clotting factors such as factors 5, 7, 8, and 9 to further amplify and upregulate the coagulation process via positive feedback. The more thrombin that there is, the more previous factors up the cascade are activated. So, what does this all mean in a clinical context? Well, one thing we quickly realize is how many clotting factors actually require calcium ions to activate them. These factors are factors 5, 7, 8, 9, 10, 11, 13, and thrombin. This would explain why some medications would target these portions of the cascade and act upon the calcium binding steps. Remember this when we enter the pharmacology section. Another notable characteristic of the cascade is realizing how one factor protein at the beginning of the cascade can amplify thousands of thrombin to promote coagulation, especially if each thrombin holds multiple procoagulation functions and create more fibrin strands for every tissue factor or factor 12. Remember, the goal is to amplify fibrin strand production in order to strengthen the clot. Being familiar with the cascade can also help us to understand why we might choose to investigate certain lab tests, administer certain anticoagulants, or how certain coagulopathies are manifested.